Good morning, everyone. I welcome you all to this 17th day CSIR online training. Before entering into the section, please confirm am I audible? Can you hear my voice? Please let me know. Types. Okay, thank you so much. We will start the section. Okay. Yesterday itself, I am discussed some important topics about the DNA replication. In this unit 3, we have replication, translation, transcription. That different fundamental process in detail we are going to study in the unit 3. Just I'm refreshing the replication because that need a correlation for transcription, so only I'm included some slides and I'm repeating this. It's in this replication process, that's a single strand is divided by helicase. This divided into two strands. After the replication fork formation, the RNA primers are added to the each added to the strand with complementary nucleotides. Then some Okazaki fragments are bind at the lagging strand. One more thing, here the questions may ask like Okazaki fragments can only bind with the lagging strand. It's a question. It's an answer. It can if there is a Okazaki fragment present in this strand, that is called as that is the lagging strand. That lagging strand only have the polymerase enzyme, DNA polymerase enzyme. This DNA polymerase enzyme helps to extend the DNA. It's all about the replication fork. This replication fork is initial stage of replication, and it's a replicon is dividing two adjacent origins preparation. Okay, I'm once again repeat how can what is the kind of repair happen in the DNA. Here this is the base excision repair. It's may caused by ionic radiation. This ionic radiation, it's DNA, it's is DNA. So the specific binding site, it's this AP binding site processing. That AT binds by AP site processing means in that particular enzyme, it deactivated by the ionized radiation. So there was lack of patches, that there was lack of the nucleotide attachment that make the repair. Okay. That After that, removing the overlap, this after that, this is the damage. After finding this damage, we can synthesis the short patches. Short patches means it's a divided, if there is a damage, once we find out the damage in particular place, we want to cut down that DNA. Then after the cutting, it may that addition of nucleotides may be the long that make up. Oh, here you can see a single or a single pink color, these are the nucleotides. It may be separately added. Okay, otherwise you can add the uh, you can add only one or you can add more. These are the repair process. You can add the nucleotides once. First cut the particular repaired damaged DNA. After the after the separation of both DNA, you can add single nucleotide to the repaired region, or you can add multiple, that is the long patches. Okay, this kind of single nucleotide addition without any other processing is called as the base excision repair, that is the BER. Another thing is nucleotide excision repair, NER. Nucleotide it can helps to repair the damage caused by UV damage. Okay. Uh, this nucleotide excision DNA repair. This repair depends on the helicase activity. Here, this helicase activity produce the fork, right? Here you can form the fork. After the fork formation, that DNAs or strand is formed. During the strand formation, it uh, may be damaged by the endonuclease. So there was some damage in the sequence. You can add nucleotides with polymerase enzyme, then it can be healable. It's a nucleotide addition of nucleotide to the strand, repaired strand, damaged strand, that is nucleotide excision repair. Then homologous recombination, that homologous recombination is a very important concept in the basic fundamental processing, because many of the questions are specific from this topic. So it's not a tedious process, but this is a usual in commonly it's divided into homologous and heterologous in different manner. If you recombinate both similar nucleotides, nucleic acid strand, if you bind or recombinate the similar species, similar natured sequence, means that is called as homologous recombination. In this homologous recombination, first step is the overlapping of two DNA strand. Here in this image you can see two blue color strand and pink color strands are there. Some portion of blue color T is region sequence, some that are divided into that are incorporated into the pink region. So here you can see the pink and blue are same sequences, but that sequences are interlinked. This is the concept here. There is it's called as the Colliday junction, that sequences are exchanged between both two different nucleotide sequence, right? That sequential changes are called as Colliday processing. That exchanged region is called as Colliday junction. Okay, next is homologous end joining. In this homologous end joining, it's nothing but adapter kind of addition. Here this non-homologous end joining is helps to repair the damage caused by the ionization radiation. After that endonuclease process in the DNA, there was some lagging damage happened in the DNA. That DNA was overlapped and that are repaired by the nucleotides with enzyme that polymerase. This is also yesterday I discussed, I think so. Here the DNA, here it is starting step is here. Agrobacterium tumefaciens, it's an bacterial system and is a plant. It is most of the plant research, molecular biology and genetic engineering researchers mostly widely doing the agrobacterium. Before going to the section, please study about the agrobacterium tumefaciens, because it's most repeatedly asked concept, that agrobacterium tumefaciens concept. Once you understand the concept means, then you can easily answer the experimental question. The agrobacterium tumefaciens concept is very very important in the CSIR examination point of view. Okay, in the recombination itself you can find the TI plasmid. Okay, in that plasmid having the TI, it's having some restriction site with specific restriction site. So if this restriction site is restricted and inserted with that region is inserted with the recombination new gene. So the formation of new gene is inserted into the competent cell. That cell is injected to the plant. It's a recombination process. So it's a plant specifically possesses the character, some specific character. This inserted gene is the responsible for specific character. These are all the specific enzymes which helps for the each and every step. Polymerase that function as a replicase enzyme. Then RNase polymerase 1, it's a repair enzyme. Ligase, that ligase helps to bind seal the sugar phosphate backbone. RNA polymerase, it's make RNA primers. Get RNA polymerase 3 started, and RNase H, it's remove the RNA primers. First one is RNA polymerase. That RNA polymerase or RNA primers are helps to form the RNA primers. RNase H, this helps to remove the RNA primers. In that exam point of view, they ask the multiple choice questions like that. So only I'll make this table, because that they are asking this in this kind of view. I am faced that this kind of questions, because DNA helicase, they provide the enzymes names and ask the functions. So in some exam point of view, we may get confusion. So prior to that, we want to understand step by step process. Okay. Here it's a very important thing as DNA helicase and chirase. It's both are helps to unwind the DNA. Okay, it's a replication fidelity. Here this in this replication fidelity, you can may have the two different reasons why it happens. That reasons are this it's depends on the damage happened on the DNA. Okay, if there is a DNA, it's damaged by any other reason, we want to get the repair the DNA. So for that process, that enzymes are activated. That mismatched pair don't have hydrogen bond properly. So there was unstable bond in the DNA. So that make the damage. And DNA polymerase active site of accommodated, its active site is only accommodated properly matched base. Its mismatched base are not not identified by the polymerase, so there is no extension, there no DNA strand formation. This is the most important thing. First one thing is that want to be stable, the nucleotide arrangement want to be stable. Another thing, that if there is a stable nucleotide in the sequence, that or only that can that can allow the DNA polymerase activity. Then this is extra chromosomal elements. Extra chromosomal element means these elements are produced from the external source. You can see here, this is the cell over the bacteria. In this virus, it's releasing their chromosomal into the bacterial cell. This is called as extra chromosomal region. This extra chromosomal region can interrupt with normal host genomic DNA. This process may happen in the recombination. If this recombination, it makes the bacteria forge. Okay, this is the step-by-step process where we can repair the mechanism. One is accession, that is we discussed earlier. Accession, the damage is cut by one of the series of nuclease enzyme, each specialized for type of DNA damage. Another one is resynthesis. Original DNA sequence is restored by the repaired polymerase. This is the second step. Restored by the repair polymerase, DNA polymerase. Another one is after the repair, that nucleotide formation, there was ligation. DNA ligase seals the nick left sugar backbones of or repaired DNA strand. These are the different three steps: accession and resynthesis and ligation. This is the what is the cause for this damage, type of damages, and how can we repair the that damaged sequences. First one is the base match mismatch. One is base mismatch. Base mismatch means there is only A and T is the correct base, but some in the due to the some causes that bases may vary. That may be the ACGT. That is the mispairing. In that mispairing, that we want to remove the particular region, and that is a mismatch repair. In the mismatch repair, we want to remove the particular region and add the nucleotide sequences for one complementary strand. In this case of mismatch sequences, they remove one strand. They remove the one strand and add the nucleotides and activate the polymerase enzyme. So we can get the perfect matched sequence in the opposite strand. Another thing is single strand breakdown. In this single strand breakdown, it's happened because of the in between bond breakdown. In that there is a bond breakdown between the nucleotides. It's if there is a and T, a bond between A and T is strong in manner, there is no damage. So this external causes make the damage between A and T and G and C. So the bond is break down. That is formed by the base accession repair system. So there is a addition of enzymes and add the both nucleotide sequences. Another one is the DNA adducts. Here you can see here the internal some unwanted bond between the same between the nucleotides, nearer nucleotides in the same sequence. Okay, in the one strand itself we have the A and T in the next manner. First is the A is there in the strand, another followed by there is the T. So that bond is mixed between the both A and T in the same strand. That is the wrong one. That kind of process are repaired by nucleotide accession repair. Okay, this nucleotide accession repair, it's after the removal you can get the sequences. Transcription couple or global genomic repair is happen in this DNA repair. Another thing is a double strand repair. It's after if there is a double strand repair means you can homologous. It's that particular part should be removed and add the additional parts. That is called as the homologous recombination. In this homologous recombination, we discussed earlier, that is made by the holiday junctions, and synthesis depends on the strand annealing. Okay, this opposite where there is a missing means you can add the take the sequence from another DNA and add it to that. These are all the causes and how outcome, what is the outcome, what is the negativity. If there is DNA is muted by the UV radiation, means that radiation is damaged effective the defective GGR, NER and TLS. These are all the enzymes in the genes in the nucleotide. So there is the higher rate of cell death that may mutate the gene that cause the genetic instability. Then this photosensitive that makes photosensitive and cancers. Okay, next is the normal with this green symbol is a normal one, normal repair, genetic integrity it's caused. So no, there is no disease. Our system is also automatically repair the repair that in nature. The next is the endogenous reagent, that may be the ion, that may be the any causative agents that defect the NCR and NER. This after that damage, it's increase the cell death. This developments the neurological problems. The some of the other diseases are also caused by this damage. Here also it's the easiest form of the explanation how it has happened. Here some of first ionization, this ion radiation cause the damage, that is that repaired by the non-homologous end joining method. So at the end alone we can change the sequence. It may be cured by the homologous recombination. Then UV chemical the damage caused by the UV, that is repaired by nucleotide excision repair. Oxygen radicals hydrolysis some compounds, that is oxaloacetates 8OXO. These are the oxygen radicals hydrolysis the alkylating agents. This kind of damages cause the A basic site single strand breaks down. This breakdown is repaired by the method base excision repair. Then replication error, it's a natural one, is mismatch pair. That mismatch pair is repaired by the this mismatch pair is repaired in an automatic manner. These are all the causes. What happened? It's a single strand, or it may be this double strand, or it may be the it's a DNA adductant. All are important thing. Here you can see ionization makes a single strand breakdown, double strand breakdown, intra-strand cross-link and inter-strand cross. These are all the causes. These are all the damage type. Radiation causes the bulky adduct and its pyrimidine dimers. Then oxygen radicals hydrolysis and alkaline agents are causing the single strand breakdown. Then replication error make the mismatch insertion. These are all the what kind of DNA. Why I am repeating this means these are all the very very important thing, that basic questions, because in this scientific world many of the researchers doing the this kind of researches. What is the damage? Because cancer, neuronal diseases, many diseases are in this world, because so researchers doing on research on that point of view only they are asking the CSIR question. um, Once you understand how it is getting denatured, damaged, how it can be repaired, and if you know this mechanism, you can easily overcome the research problems. So only I am asking this kind of, they are asking this kind of rapidly in the CSIR examination. This is it's a normal kind of recombinations, homologous recombination. Homologous recombination, as I said, two different sequences, nucleotides are incorporated with the specific regions. Another one is site specific recombination, then DNA transposition. DNA in this here another site specific recombination. We have three types: insertion, deletion, inversion. In this in this site specific, you can see the insertion. You can insert the new DNA that is recombinant into the particular region. The AB gene is inserted into the a region, specific region of X and Y. Here you can the deletion. After once you have the whole DNA in the site specific recombination, you can remove the specific AB alone. And then it's inversion. You can invert the DNA sequence, the recombinated sequence during the experiment. Okay, next is till now we discussed about the what is the DNA replication, what is during the DNA replication we may face the DNA repair, how it is can be DNA damage is caused during the replication, and it can be caused by the reagents. How can we repair the that damage? These are all we discussed. What are the enzymes involved in the DNA replication? These are the basic concepts. Next we are going to discuss about the basic transcriptional mechanism. In this transcriptional mechanism, we are having what is transcription. In this transcription, the DNA it's it's converted into the RNA sequences. This RNA sequence conversion is called as the transcription process. Yesterday itself we discussed in the Opron concept, that Opron concept in that DNA is that particular gene producing the mRNAs. That is the concept, that's basic trinary. In this, the DNA dependent RNA polymerase. You may hear this word in the this pandemic because the main enzyme of viral production, replication, cell growth. The viral growth depends on the RNA dependent RNA polymerase. It's only produce the proteins for the viral assembly. If you stop the protein assembly, you can control the viral infection. So this transcription, that is the mRNA production. RNA production is the major role playing concept in the transcription. So there is the RNA dependent RNA polymerase. This RNA dependent RNA polymerase and then it's playing a vital role in the protein production, because the protein production, that mRNA is initial. So if we stop the mRNA production, we can automatically control the protein production. This is the concept. Then is a core promoters. This growth promoters on the DNA is used to position the RNA dependent RNA polymerase. Another some of the regions are there, that is the enhancer, silencer, stimulator. There are the some concepts. Then transcription initiation mechanism. What we want to start this transcription, we want the signals, right? This signals can be passed by the promoters, activators, enhancers. That is the some of the initiators. You can understand the concept now. Yesterday we discussed some lacopron and trypopron, right? In that trypopron, you can it's the if that promoter region is bind, that is produce the that make the signal lagging in the signal. If there is a lacopron, if you bind the lactose in the gene, that promotes the signal to the gene. These are all the some concept understanding. Okay, that initiation signal is formed through the that complex. That okay. Oh, what are all the compounds in the that initiation complex? This you can see: chromatin remodeling complex, mediator, histone acetylase, transcription. This factors. Okay, this are all the very important thing. Transcription initiation factor. This activator protein bind with enhancer. That enhancer is the binding site of activator protein. The next important thing is binding of general transcription factors, that is the RNA polymerase, mediator, chromatin remodeling complex, histone acetylase. This histone acetylase very important concept in the this protein mRNA synthesis. That is a transcription initiation machinery. Okay, it's one other here I am include some images for understanding. Here you can see the insulator, that is the insulator, diacyl, that is enhancer. Then upstream process you have enhancer, then silencing. Here you can see some genes, then downstream process. Some specific regulatory elements are there in the initiation process. These are all about the initiation complex, initiation things. Here you can docking site for site specific regulatory protein. Site specific regulatory protein, that is the this transcription factors, TA binding factor, and site specific binders. These are all the specific transcriptions factors. This they are having the specific docking site, that is a binding site or there in the DNA. Otherwise it won't activate it. That initiate the transcription process. These are all the factors are necessary for on the over the DNA. Then enhancers, tissue specific activators of transcription. Tissue specific means for each tissues, each proteins we have a specific activators. Then silence, and after finishing this process we want to silence or enhance the transcript. It may be enhance or silence, that depends on the production and signal rate. That is the prevalent. It's a this makes the gene, it makes gene expression perfect manner. So we want to balance the gene expression. There is a limited amount that may if there is need you want in production, other no need. If there is no need in the particular protein production, we want to stop the production. That depends on the that enhancer and silencers. Then insulator. This insulator is the boundary element. It shield gene after particular region in the heterochromatin protein. It's a heterochromatin. You can see it's a chromatin in the chromatin or DNA. You can see particular region for particular gene production. After this insulator is a bound. This is the region. It's you can see this a dicyl region. It's nearly it's in one kind of two more question. Dicyl region having 200 base pairs in upstream. Proximal region having 50 to 200 base pairs, and core region having the 40 to 50 base pairs. Okay. In this this dicyl region having the NRE, that is non recombinant that repair regions. You can see here. Okay, yeah, any NRE. Then this is the central. I'll check it and tell you this what kind of in the image, because it's not visible to you. I will send the proper image. The presence of the genes are very important. This NRE gene present in the dicyl region, gene is present in the proximal region. Then core region only having BRE, and the TATA gene, INR gene, that is the insulator gene, and the CPE gene. These are all the important things. These are all the gene names. In this in the transcription initiation process itself, these are all the major regions which involves in the process. This next one is the elongation process. After initiation of the transcription, here you can see the strand that site is formed. It's identified by TFIT IAT. This is the major component to form the initiation complex. After the initiation that binding, that's TF compounds, or TF11 IA, or the IAF, IAH, IAI. These are all the bound at one particular bound at one particular place and make a complex. Then RNA polymerase being elongation process, start the elongation process. So this strand, this RNA polymerase, is specifically add the phosphate groups and to form the new strand from the DNA, from the RNA. That is the RNA polymerase 2 being elongation. So another one example here I mentioned, as I am already told, I am included different type of images for understanding. First one is the TATA gene region. That region, this TF2 T going to bind, that they mention the last letter alone. The yellow color having the D. This D is bound with A. Then A and D is form a complexion with B. This ADB complex is attract the F. Then F, this ADBF complex bind with H and E. These are all the step by step process. This may be the question. What is the order of binding of complex, initiation complex? G, then A and D, then ADB, then ADBF, then ADBF EH. These are all the sequential process. So here they mentioned repeated that the flow chart is in word format. The enzymes used in the particular step, that the binding of TF complex with RNA polymerase complex binds the B. F can bind with B by the RNA polymerase enzyme. F and E is open strand, it's helicase and ATPase activity. Then activator and repressor. After the initiation complex formation, we want to activate the replication or reduce the process, stop the process. That is called as the activator and repressor. It is specific bind to the psi element, specific psi element. This promote or inhibit the assembly of transcription. Okay, that only pass the signal to the other genes. These signals are recognized by the RNase polymerase. Okay, we want to bind or not. That understanding is stimulated by the activator protein. Here, the process of activator and activators. Okay, here these are all the pieces of different amino acids. These pieces represent different proteins. These proteins are bound and forming the complex that is called as the co-activator. This co-activators the activates transcription process. Co the expressors are repressors. Co-repressors are repress the transcription process. Repress means it's off, stop the process. Activators is a gene on process. Okay, this is a very important concept. Here's what is the role of repressor means gene to produce a transcript. That transcript is mRNA. Then that if co-repressor is added to particular gene complex that stop the mRNA synthesis. And another concept is addition of that cofactors and addition of the co-activators. If there is addition of co-activators in the DNA sample, that makes the start the transcription process. Then this is the where this is the activator types. These are the genes of activators. This RNA polymerase 2 complex and that enhance binding of proteins and mediate the signals between the both complex and DNA. Then RNA polymerase 2 may carry the co-activator protein. It's a transcribe. It is acid transcribe. Then co-activators are necessary for the transcription, so the RNA polymerase carries the co-activators. Here you can see this image. This is the co-activator bound with the activators. Here this this whole complex, it's a having enhancer, DNA binding domain, and having the promoter region. It's all in the one particular DNA loop, the particular region having all those things. This here you can see this is a full strand. If you make a addition of co-activator means that strands forms like this. That formation of this cofactors, TF factors are transcription factors are forming in the complex. There was gene production. Otherwise, that there was the elongations happen. Otherwise, there is no elongation that makes some genetically modifications in the transcription process. So activation of co-activators is very necessary for all the transcription process. Then in this elongation process, the RNA polymerase play a major role. Why we need the RNA polymerase? This RNA polymerase, it's unwind the it catalyzes the formation of phosphodiester bond between nucleotides. So this RNA polymerase, it stop the that phosphodiester bonding. So uncoil the DNA. This is a very important thing. Here you can see the image. This orange color is the DNA. If they using the RNA polymerase, that single strand is transcribed in the from the DNA. That unwinding, short region formation, then unwinding, rewinding. This first one is unwinding, then short formation, that is then one is the rewinding. These are all the three different steps in the RNA polymerase activity on the DNA. Then function of RNA polymerase are it's search and bind to the specific protein and unwind the start sequences and ribonucleotides formation, then detect the terminal signaling, then stop the process. The main thing is it forms that catalyzes the formation of phosphodiester bond. It's a very important thing. Then what is the kind of RNA polymerase? It's a types classification. We discussed some DNA polymerase in the earlier stage, right? Apart from that DNA polymerase, there is a different types. RNA polymerase 1, 2, 3. Likewise, here we have the RNA polymerase. In the replication process, we discussed the DNA polymerase. In the RNA transcription process, we are going to discuss about the RNA polymerase. The RNA polymerase is the key role playing component in the transcription process. So here it is classified into 3 types based on their sets with many subunits. That only the large ribosomal RNA subunit precursors, that is the recognized by RNA polymerase 1. If there is a pre-mRNAs or RNAs are formed, means that are by RNA polymerase 2. We want to produce transcribe the tRNA, 5S rRNA, U6 single SN rRNA, that 7SL are all the types. Okay, these types of RNAs are produced that are activated by produced by the RNA polymerase 3. Then mitochondria and chloroplast in itself, they having these RNA. The RNA polymerase are makes a subunit, single subunits and multiple subunits. In a eukaryotic, RNA dependent RNA polymerase is a very important thing. This some large virus it's encoded by the own polymerase itself. That is called as 3 different type of different organisms that are having different polymerase, different activity. It's another complex how it is arranged. Okay, it's a very important thing. If for RNA polymerase 1 is activated by the different factors, that are different factors genes, that is the UBF, the TA activator region, the transcription activators, and it's 47S pre-RNA. Only it's developed by this RNA polymerase 1. Then another thing is for RNA polymerase 2 activation, it needs mediator complex. This mediator complex MED26, and this can extend the TATA region. So produce mRNA, snRNAs and scRNAs. Then another one is RNA polymerase 3. This RNA polymerase 3 is we discussed the normal transcription process. That is need the cofactors alone. That is the transcription factors. After binding of transcription factors, it's activated and develop the transcribe the DNA. It can help to produce the tRNA, 5S RNA, SN RNA, scRNA. We want to understand why we need the RNA polymerase. They provide the experiment. Okay, you need to develop the snRNA. For snRNA, what kind of RNA polymerase you need to add? That kind of questions you need to answer RNA polymerase 2 and RNA polymerase 3. One more thing, in the many CSIR question, they are given 2 or 3 answers in the option. Okay, A, B, C, D. Here the answer is A, B, C. In the question itself, they provide the option A, B, C, A, B alone, A, C, C alone. This kind of answers are there. So please understand, if there is a question like SN RNA production, how can we synthesis. What is the enzyme used in the snRNA synthesis means you need to answer. You can add the polymerase 2 or you can add the polymerase 3. For tRNA, polymerase 3. 5S RNA, 5S RNA you need to add a polymerase 3. For 47S pre-RNA you need to add the polymerase 1. These are all the basic concepts. Then for miRNA we need to add polymerase 2 and polymerase 3. Okay, this RNA polymerase present in polymerase 1, 2, 3 are present in eukaryote, polymerase 4 and 5 present in plant alone. This is another thing. Polymerase 4 and 5 present only in plant. These are some transcribe the siRNA. Polymerase 4 helps to transcribe siRNA. Polymerase 5 helps to transcribe RNA molecules, take part in the heterochromatin formation. Okay, next concept is cap synthesis. It's a after the RNA synthesis, RNA strand formation, we want to synthesis the cap synthesis. The first step is RNA triphosphate. It's removed from the terminal phosphate from pre-mRNA. That is the before the initiation process to capping GMP. It's capping. It forms the GMP from the GTP. You can see here it's a very important concept. There is the pre-mRNA is there. We want to add RNA triphosphate. This addition of it helps to form the complex B. That is called addition of gluconyl transferase. It helps to modulate the addition of the gluconyl transferase of G to the sequence that forms one complex. That complex is activated by methyl transferase, then addition of methyl to the sequence, then there was again add the methyl transferase that makes a multiple sequence. It's a capping process and splicing process. In the capping process you can add the capping enzyme and form the multiple sequence, form the cap at the end 5 prime. It's a sequential process. You can see here it's a very well explained image. It's a nucleotide sequence. It's a 5 prime to 3 prime. You want to add the phosphate, that is the previously itself is triphosphate, RNA triphosphate. You added that RNA triphosphate that forms the GTP GTP and release the PI. Here you can see the one phosphate group is removed. Here you can 3 phosphates, that is a triphosphate, that is phosphate addition of the phosphatase enzyme. There is a removal of P. That also you can see the only 2P. After the addition of gluconyl transferase is added to the GP. Here you can see the GPP, NP and NP. That addition of methyl group to the bases. That methyl group is added by guanine 7 methyl transferase. This guanine 7 methyl transferase is added to the end of 5 prime N. This is the methyl CH3 addition to the GP. Then methyl transferase those is capped with. You can see again I had the methyl transferase 2O methyl transferase that make a cap. Here you can see here you added the methyl. It's a one kind of post-translational modification. This after the MR happen in the cell, in the DNA transcript, that RNA transcript. This modified guanine is added, right? That is the main thing. We are added the guanine. It's a one important thing happen in the capping. After the addition of guanine, we are closed that region with methyl. The why we need to protect the mRNA from degradation. Degradation means once if there is any enzyme is found the guanine, that starts makes a degradation. To avoiding the degradation, we are capped, we are closed the particular region. So if produced transcribed, it's at the end we are added the phosphate group by RNA that triphosphate the RNA triphosphate, this RNA triphosphatase is helps to remove the last ending P. This P is removed, then guanyl transfer is added. This guanyl transferase addition is makes GP, NP, NP sequence. That's GP is capped with methyl transferase enzyme. So methyl is added with the methyl transferase, then added the 2O methyl transferase. It's form the bonding with the first P. It's very important thing. First you have added guanine 7 methyl transferase. Second step again for methyl base 2 ribose, you have added the 2O methyl transferase. So one is methyl group added to the base, that is the guanine. Methyl group added to the guanine, that is a base. That in that case they used guanine 7 methyl transferase. For next case you have to add the methyl transferase O. It helps to add methyl to the ribose. It's a kind of post-translation. This is again I am explaining for understanding. I am added the image. This is the elongation process we discussed earlierly, that RNA polymerase play a major role. This how the transcription happen in the eukaryote. This promoter region is having some region, that is a data box region. This is data box region is 25 to 30 base pairs in upstream. This upstream region is recognized by the transcription factor 2B. After that the complex is formed, then that complex is helps to make a transcript. Elongation process is a similar process. Here you can see initiation, that complex formation, and elongation, that individual phosphate groups added to that particular region. Here this is the that intermediate portion. In that region we have 3 steps. I already discussed in the previous slide, right? That RNA polymerase addition, there was some unwinding, then after the unwinding the addition of the phosphate groups and forming the DNA strand, the RNA strand, then rewinding of DNA, then termination. After the formation we want to terminate. This transcription termination, it's 2 mechanisms are there, that is a row dependent mechanism. In this row dependent mechanism you can form it, that is a hairpin loop formation, then that hairpin loop formed followed by the stretch of U residue. Okay, otherwise there is no interaction. If there is a U in the strand, there was the loop, hairpin's loop formation, otherwise there is the straight line. The row dependent mechanism, the termination signals sequences are transcribed there itself alone, then hairpin forms followed by stretch of U residue, then stem loop cause the RNA and RNA polymerase to pass in that stem. Once there is a stem is formed, means okay, that RNA recognized, okay, there is no need for the synthesis, that is enough, that in that gene is terminated, that gene is that insulator make a sense. Okay, next sequences are responsible for another another mRNA, so no need to develop. This is the basic concept. This is all about the RNA processing. In this RNA processing, we have two different process. One is pre-RNA transcript, that is the 40S RNA. Here you can different sequences that after the methylation, some unwanted regions are recognized. Okay, these are all the regions having methyl. So methyl having regions alone is known as the coding regions. So that are all the excess regions are cleaved, and now we have only the coding regions. Okay, we can modify the RNA by the RNA editing. If there is if you want to change the mRNA, that is based on the alteration of amino acid sequences, then altering the future pathways, then alter the splicing sites. Then another one is a function based one, DNA degradation and DNA replication. These are all the function based editing. In the function based editing, have DNA degradation and DNA replication. Another editing is DNA structure based edition. RNA structure based edition. This RNA structure based edition, we have the secondary RNA structure modification, that is the protein binding site based on the sequence change, that is secondary sequence change. So there was different type of editing in the RNA editing. In the editing mechanism, it's a cleavage, means deletion editing means just to find out one region and remove particular the sequence, that is the deletion. After the deletion, we want to form the new sequence. This is the deletion editing. Then insertion editing, one region is it's very needed one, means unwanted things are removed from particular place, then add the addition of U to the particular region. Here in this step we have to understand, for cutting off particular U region by endoribonuclease, that cleavage is caused in the RNA editing is caused by endoribonuclease. That's a very important thing, endoribonuclease. Then U sequences, removal of removal of U sequences is caused by exouase. Another one is the addition of U, it's a TUTase. After the addition and deletion, we want to close. For the close, we want to add the RNA ligase. It's RNA splicing. In this RNA splicing itself, we have the same process, that extra non-coded regions are removed, and it's spliced, and mature RNA is produced. That on-coding regions are removed, that is a splicing process. This is the splicing spliced RNA. Here cleavage of the another one concept is the polyadenylation or cleavage, the final concept in the RNA technology. That polyadenylation sequence, there was a sequence that endonuclease enzymes, it helps to cleaves are 20 nucleotides in downstream from that particular AAA sequence. Why we need to remove that? If we want develop the particular poly A tail, we need to remove at the some sequence at the end, that RNA poly A polymerase. It's added adenine nucleotide at the 3'. First step is polyadenylation. Poly means multiple. 10 means it's a addition of A at the one end. Addition of one similar nucleotide. So first step in the polyadenylation sequence, first step we have the endonuclease to cleave occur at the 20 nucleotide at the end from the downstream. Where this not happen at the random. Once the enzyme found AAUAAA, this 5 nucleotide in the DNA sequence, that recognize endonuclease is okay, it's understanding, okay, this is our site, we want to cut 20 nucleotides after to this sequence. So this after to the AUAAA sequence, to after 20 nucleotide of this particular code ring region, that polyadenylate that endonuclease is added to the DNA and cut down the particular region. That is the cut happen here. This is the nucleotide 20 nucleotide downstream. That is a back to the 3' end. And then another is poly A polymerase that makes the addition of A. This is another example. Example, not the imagine. It's a pictorial representation for cleavage of how it is in the happen, adenylation, how it is happen. So another pictorial representation. Okay, what is the types of RNA? Types of RNA, it's a transfer RNA, ribosomal RNA, the messenger RNA, small nuclear snRNA, small interfering RNA, that is the snRNA, siRNA. This transfer RNA is very, very small. It transport amino acid to site of protein synthesis. Then ribosomal RNA, it's a several kind of variable in the size that combine with the protein form the ribosome. In the site of protein synthesis. Then messenger RNA, it's a variable size. It's a direct amino acid sequence of proteins. Then small nuclear RNA, it's very, very important thing. Small nuclear RNA, it's a process the initial mRNA to mature mRNA formation in a eukaryote. At a small nuclear RNA, it helps to get the mature RNA in the mature RNA in a eukaryote. And small interfering RNA helps to affect gene expression. Knockdown process, they are using siRNA. Very, very important concept. If the researchers are doing the knockdown gene experiment, they are added the siRNA, that is the small interfering RNA. That microRNA, it's small RNA, it's affect gene expression, it's important for growth and development. Here it's a structure of 3 different mRNA. Here, because based on that kind of only they may ask the question, so only I am included. If there is a messenger RNA, means that is a very small one, it's a single stranded one. Transfer RNA, tRNA, we discussed the tRNA structure in earlier classes. Then ribosomal RNA, it's a group of 1. Today we are remaining part, that is the protein synthesis process, the translation process, and gene expression control in phage, virus and prokaryotic. Tomorrow we will discuss this kind of topics, remaining topics of unit 3. Thank you everyone. Thank you for your patience. Please post your queries in your chat box.